Good afternoon. I am the expert, Sergey Somov. I continue to introduce you to MAGUS microscopes. Today, I will talk about the inverted biological microscope MAGUS Bio V300, which is used in clinical cytology, biotechnology, virology, agriculture, and other fields of science that study living organisms. The microscope is used for education in universities, routine laboratory work, and scientific research. Inverted means turned upside down. Such microscopes were invented for the study of living objects in special laboratory glassware. The microscope works with petri dishes, vials, roller bottles, and plates. It is also suitable for studying samples on microscope slides. Although for such research it is better to choose direct microscopes. Inverted biological microscopes are used in scientific institutions, microbiological laboratories, and IVF clinics. You may have seen beautiful staged photographs where a petri dish with colorful mold sits on the table of a direct biological microscope, while a pretty girl looks into the eyepieces. In fact, a direct biological microscope is not suitable for working with glassware. Firstly, the objective of a direct microscope cannot focus on the object. The lid of the petri dish will obstruct it. Even if we open the lid, we will encounter a new problem. We will have to immerse the lenses in a nutrient broth with live microorganisms. It is better not to do that. Secondly, the illumination system of a direct microscope is designed only for working with flat preparations on thin glass slides. The condenser will not focus light on the sample through the bottom of the container and the culture medium. Thirdly, purely mechanically, a tall dish may not fit into the microscope. Therefore, to place a petri dish or another laboratory vessel with a research sample on the microscope stage, a microscope with an inverted design will be needed. Look, we place the petri dish on the table. In the dish, bacteria have been multiplying on the nutrient medium for a week, forming colonies and living their everyday lives. I don't know what has grown there, whether they are pathogenic or not, so I securely sealed the lid of the petri dish just in case. The objectives are located under the stage, while the light source is positioned above the stage. Light enters the condenser, passes through the specimen, and then reaches the objective. The objective forms an enlarged image of the object. Mirrors direct this image into the eyepieces. I have explained to you the principle of operation of the inverted biological microscope. Now let's take a closer look at the specific model MAGUS Bio V300. The visual attachment is trinocular. A digital camera is installed in the vertical tube with a visualization channel. The division of the light beam is constant, without switching. 50% of the light goes to the eyepieces, and 50% to the visualization channel. Thus, you can observe the object of study both through the eyepieces and on the monitor simultaneously. Type of visual attachment: Zeidentope. The tubes rotate smoothly around the hinge for easy adjustment of the interpupillary distance, which ranges from 48 to 75 mm. The range is wide, making the microscope suitable for both adults and teenagers. The standard equipment includes 10x eyepieces with a field of view of 22 mm. The diopter adjustment of the eyepiece helps to customize the diopters to the user's vision. The eye cups protect against side light. The revolver with 4 objectives is located just under the table. The revolver features planochromatic objectives for infinity. The objects of study are housed in special containers, which is why both the stand and the optics of the microscope are designed to work with this glassware. The microscope uses objectives corrected for a bottom thickness of 1.2 mm. 3 objectives with magnifications of 10x, 20x, and 40x operate using the bright field method, and one 10x objective operates using the phase contrast method. Additional equipment may include a 4x objective and phase objectives of 20x and 40x. The planochromatic objectives correct spherical aberration for 1 wavelength of light, chromatic aberration for 2 wavelengths, and correct field curvature by 90%. When used with eyepieces that have a field of view of 22 mm. The MAGUS Bio V300 microscope operates using bright field and phase contrast methods. The condenser has a slot for installing a phase contrast slider. When working with a phase objective, we introduce a slider with the corresponding phase ring into the path of the rays. The phase rings are centered. During operation using the bright field method, we introduce the beam of light into the free aperture. The use of the slider saves time when switching from one research method to another. Focusing. The coaxial knobs for coarse and fine focusing are positioned on both sides of the stand for the right and left hand. Focusing on the object is achieved by moving the revolving nosepiece with objectives. Fine focusing is required for precise focusing on the specimen and for refocusing the microscope when changing objectives or laboratory glassware. The coarse focus locking mechanism is used to speed up the readjustment of the microscope when changing the object of study. The mechanism for adjusting the stiffness of the coarse focus will help set a comfortable stiffness for each user. Another parameter of the focusing mechanism is the division value of the fine focus. On our microscope, it is 2 microns. For a student and routine microscope, such a step is optimal as it ensures both speed and accuracy of focusing. Stage. A special mechanism is installed on the stationary stage for moving laboratory glassware in 2 mutually perpendicular directions. The mechanism moves the cup smoothly so that the user does not miss any part of the specimen being studied. The movement handle is long, so the user will not strain the wrist of their right hand. The microscope comes with 3 holders for different sizes of laboratory glassware. Light source. The 9W LED brightly illuminates the object. The intensity of the light is sufficient for both bright field and phase contrast methods on all objectives, even though the light flux is divided between the visual attachment and the visualization channel. Unlike a halogen lamp, the color temperature of an LED does not change with variations in brightness. The lifespan of the LED is 50,000 hours. The compact stand has a convenient handle for carrying. You won't have to think about how to grab the microscope more comfortably to move it from one place to another. We offer a range of accessories for the microscope. Additional phase objectives of 20x and 40x are used for identifying non-contrasting objects measuring 0.7 microns and 0.4 microns. Eyepieces expand the magnification range of the microscope. An additional pair of eyepieces will help achieve useful magnification on the objective, which is more commonly used for work. Digital cameras display the image from the microscope on a monitor and save the information as files, while the software measures objects in real time. The calibration slide for measuring objects works in conjunction with an eyepiece with a scale or with a camera program. The MAGUS BIO V300 microscope is convenient to use and easy to handle. Come to our demo room, bring your samples, test it out, and we will help you choose additional accessories and a camera. See you soon, colleagues.